Hello, fellow conference attendee. My name is Shalaw Sallah from the University of Manchester, and our latest work is entitled Using Structural Analysis to Clinically Interpret Missense Variants, X-linked Genes as an Exemplar. And I would like to take a couple of minutes introducing it. Missense variants in the protein coding regions are the cause of many rare diseases, and the clinical interpretation of these variants is difficult. So improving their interpretation can lead to an increase in the rate of diagnosis and lead to personalized treatment management. Due to lack of sufficient evidence, however, a large proportion of these variants end up being classed as variants of unknown significance, or VUS. And so this is essentially an inconclusive lab result. This is where in silico tools or prediction tools can help by providing supporting evidence for pathogenicity. So identifying prediction tools that are accurate can improve variant interpretation, leading to more informed clinical decision making. So the problem is discordance and inaccuracy among the prediction tools, and this means that the clinical guidelines do not prioritize their use. Therefore, developing accurate tools for subsets of genes can lead to higher rates of diagnosis. The aim of this study was to replicate the accuracy of a previously tested protein-specific approach to variant interpretation in a number of X-linked genes, and to show this approach's predictive power through the model PROSPER, or Protein-Specific Variant Interpreter. So to do this, we pulled missense variants from different databases for all X-linked genes listed in the HGMD databases, or the Human Genome Mutation Database. For these genes, the 3D structures were identified, or the homologous models were produced when possible, which were then used to carry out the analysis of the missense variants. We filtered the genes for those that had a protein structure or those a homologous model for which a homologous model could be produced, with a minimum sequence coverage of 50% of the protein, as well as having a sufficient number of missense variant, which we set at 70 based on our previous work. So these are the two filters that we applied to the genes. We tested a number of variant features or characteristics using the protein structure or the homologous model. And here is a list of the main features included in this study. We have conservation, changes in physicochemical properties, for example, whether variants cluster in the protein, and so on. So these features were used to train the classifier models for each protein or gene. Also, when evaluating other prediction tools, we identified gene-specific pathogenicity thresholds to improve their performance. And this was done using repeated 5-fold cross-validation with random subsampling. So on the other side of the poster, we have reported the results of the performance of PROSPER, the protein-specific variant interpreter, in comparison to the best performing tools out of a group of 11 tools considered in this study. And we show, using the Matthews Correlation Coefficient test, or MCC, that PROSPER outperforms REVEL and VEST4 in 13 out of the 21 genes studied. Further down in the table, we show improved performance in prediction in a number of genes using a gene-specific threshold rather than the 0.5 threshold, which is shown in the column titled Original MCC. There are decreases in the level of performance in some genes, and this is mainly due to the effect of the 5-fold cross-validation on datasets with a smaller number of variants, amongst other reasons. The optimized MCC results are reported with a standard deviation. This is in comparison to the original MCC results for each gene for both tools, REVEL and VEST4. To conclude, we found the consensus-based prediction tools, REVEL and VEST4, are more accurate in predicting missense variants in the 21 X-linked genes. Furthermore, PROSPER outperforms REVEL and VEST4 in missense variant prediction in these genes. Also, using a gene-specific pathogenicity threshold can improve the performances of REVEL and VEST4. Overall, PROSPER is robust and can form the basis of a family-specific prediction tool which can be implemented into diagnostic strategies to more accurately prioritize variants impacting X-linked diseases. This can facilitate precise and timely diagnosis in the clinic. Thank you for listening.